All right, buckle up everyone, because today we're diving headfirst into the fascinating world of neuronal activation.
That's right. We're talking about the electrical and chemical processes that literally make our brains tick.
And I hear we're going all the way down to the molecular level.
Absolutely. We'll be exploring those intricate mechanisms that drive everything from our thoughts and actions to our emotions and memories.
Sounds complex but fascinating.
It is. But we'll break it down step by step, making sure everyone's on board. And to top it off, we'll even connect this all to the ever-evolving world of cannabinoid pharmacology. You know how compounds like THC and CBD actually interact with our brains?
No, but that's something I'm really curious about. Where do we even begin with such a complex topic?
Well, a great starting point is understanding a neuron at rest. Think of it like a tiny battery, just brimming with potential energy, waiting for the right signal to unleash it.
So it's not just sitting there doing nothing, it's primed for action.
Exactly. And this potential energy is all thanks to a difference in electrical charge across the neuron's membrane. It's like a barrier, you see, separating the inside from the outside world. And that difference in charge, we call that the membrane potential.
Okay, so it's like a tiny electrical tension just waiting to be released.
Precisely. Now, in its resting state, the inside of the neuron is actually more negatively charged compared to the outside. And it maintains this negative charge thanks to these specialized channels that control the flow of ions in and out. Ions are just tiny charged particles. These channels are super picky about which ions can pass through.
So it's like a microscopic border crossing, with those channels acting as gatekeepers.
A fantastic analogy. And this careful regulation of ion flow is what maintains that all-important negative charge inside, giving the neuron its potential to fire.
Okay, I think I'm starting to see how this resting state is actually setting the stage for some serious action. But how does that potential energy actually get released? What triggers the neuron to, well, fire?
That, my friend, is where depolarization comes in. Imagine a sudden influx of positively charged ions rushing into the neuron, like a crowd bursting through the gates of a stadium.
So that peaceful balance we talked about gets completely disrupted.
You got it. This influx of positive ions, mostly sodium ions in this case, makes the inside of the neuron less negative. And eventually, it hits a critical threshold, like flipping a switch, and bam! The neuron unleashes an action potential.
Action potential.
Yeah.
That's the rapid electrical signal that zips down the neuron's length, right?
You got it. And this is how neurons communicate with each other, transmitting information throughout the nervous system. It's incredibly fast. Much faster than I can talk, that's for sure.
I can imagine. But what happens after the neuron fires? Does it just stay in that excited state?
Not at all. It needs to reset, go back to its resting state so it's ready to fire again. And that's where hyperpolarization comes in. It's like the neuron taking a deep breath and chilling out after all that excitement.
Yeah, how does it do that? Does it just kick those sodium ions back out?
It's a bit more intricate than that. While some sodium ions are pumped out, potassium ions play a key role here. They flow out of the neuron, making the inside even more negative than its resting state for just a brief moment.
So it's like the neuron overshoots its target, going a bit too negative before settling back down.
You got it. And that's crucial because it prevents the neuron from firing again too quickly. Ensures that signals are transmitted in a controlled manner, you see. Imagine what would happen if neurons just kept firing uncontrollably.
Chaos.
Exactly. So this hyperpolarization is like a safety mechanism, ensuring that the signals are clear and distinct.
It's like a refractory period, a brief timeout.
Exactly. Now imagine if we disrupted this delicate balance. What if the neuron's bombarded with constant excitation, like being stuck at a rock concert that never ends?
My ears are ringing just thinking about it.
Yeah.
But seriously, what happens to the neuron in that situation?
That's when we run into the problem of excitatory toxicity. Too much excitation can actually damage or even kill the neuron. Think of it like an electrical circuit overloading and short-circuiting.
Okay, I'm definitely starting to see how too much of a good thing can be a bad thing in this case. But how does that damage actually occur? What's going on at the molecular level?
Well, one of the main culprits is calcium, an ion that plays many important roles in the cell. But when there's too much excitation, calcium can flood into the neuron, triggering a cascade of harmful events.
Calcium. But isn't calcium good for us? Like for strong bones?
It is. But inside a neuron, excessive calcium can be a real troublemaker. It can disrupt the neuron's internal machinery, leading to damage and dysfunction.
So it's like a dam breaking, and that flood of calcium wreaks havoc inside the neuron.
That's a great analogy. And unfortunately, this excitatory toxicity is implicated in various neurological conditions like epilepsy and stroke.
Wow, this gives me a whole new perspective on how delicate and intricate neuronal signaling really is.
It is, isn't it? But it's also incredibly fascinating. And we're only just scratching the surface. Now, imagine if neurons were just static entities, never changing or adapting. We wouldn't be able to learn or form memories.
That's a scary thought. So how do they change and adapt over time? Do they actually grow and form new connections?
That's a fantastic question, and the answer is yes. They can form new connections through a process called spiniogenesis.
Spiniogenesis sounds a bit like a dinosaur. But seriously, what is that?
Imagine tiny little protrusions, like little fingers, emerging from the dendrites of neurons.
Dendrites. Remind me what those are again.
They're the branch-like structures that receive signals from other neurons. And these little protrusions are called dendritic spines. They're the sites where synapses form.
Synapses, right? The junctions where neurons communicate. So spiniogenesis is basically creating new communication hubs between neurons.
Precisely. And as we learn and experience new things, our brains literally grow, sprouting these tiny dendritic spines, forging new connections.
That is so cool. So every new skill we learn, every new memory we make, it's reflected in the physical growth of these tiny spines.
Exactly. And what's even more fascinating is that the shape and size of these spines actually influence how strong the connection is. Larger spines with more surface area allow for stronger connections, making it easier for those signals to jump across.
So it's not just about the quantity of connections, but also the quality.
You got it. And this remarkable ability of the brain to change and adapt its connections, we call that neuroplasticity.
Neuroplasticity. I've definitely heard that term before. But can you remind me what it means exactly?
It's the brain's incredible ability to reorganize itself by forming new neural connections throughout life. It's essential for learning, memory, and even recovering from injury.
So like our brains are constantly rewiring themselves, adapting to our experiences and shaping who we become.
Exactly. And spiniogenesis, that process of forming those new dendritic spines, is a key part of this ongoing remodeling project.
This is truly mind-blowing. Before we get too carried away with the wonders of neuroplasticity, I'm really curious how all of this connects to those cannabinoids we mentioned earlier. How do THC and CBD actually influence these intricate processes we've been discussing?
That's a great question, and it takes us into the fascinating realm of cannabinoid pharmacology. It turns out our bodies naturally produce molecules that are very similar to those found in cannabis.
You're kidding. Our brains make their own version of cannabis.
Not exactly cannabis, but very similar molecules called endocannabinoids. And they play a crucial role in regulating a wide range of physiological processes, including, you guessed it, neuronal activation and neuroplasticity.
Okay, so it's not just about getting high. These cannabinoids, both the ones our bodies make and the ones we might consume, are actually tapping into a complex system that's already at play in our brains, influencing how our neurons communicate and adapt.
Precisely. And that's where things get really interesting, because these plant-derived cannabinoids can interact with this system, often with more potent effects than those naturally occurring endocannabinoids.
Okay, so it's like they're speaking the same language, but with a different accent.
Hmm.
I'm definitely starting to see how these compounds can have such a wide range of effects. We've covered a lot of ground here, but I can already tell this is just the tip of the iceberg.
You're absolutely right. And in the next part of our deep dive, we'll explore how those cannabinoids are actually influencing these intricate processes we've discussed, like LTP, synapse pruning, and even receptor trafficking.
I'm already hooked. Can't wait to hear more.
So let's pick up where we left off and delve into how cannabinoids actually influence those intricate processes we talked about. Things like LTP, synaptic pruning, the stuff that really shapes our brains.
Right, those processes that allow us to learn, adapt, remember, all that good stuff.
Exactly. And it all starts with understanding that the endocannabinoid system isn't just some isolated system doing its own thing. It's deeply integrated with all those other processes we've been discussing. It's like a master conductor, subtly influencing the entire neuronal orchestra.
Okay, I like that analogy. So it's not just a separate system, it's actively participating in the whole symphony of neuronal activity.
Precisely. It can adjust the sensitivity of neurons, make them more or less responsive to signals.
Okay, that makes sense. But I'm still a little fuzzy on how those cannabinoids actually exert their influence. We talked about them binding to those CB1 receptors, but what happens next? What are the actual consequences of that binding?
Well, let's revisit one of the processes we discussed earlier, long-term potentiation, or LTP. Remember, it's like strengthening a pathway in the brain, making it easier for information to flow along that path. It's essential for learning and memory.
Right, LTP. We talked about those NMDA receptors and their role in LTT. So are cannabinoids messing with those NMDA receptors somehow?
Not directly, they don't bind to those receptors themselves, but they can indirectly influence their activity by modulating the release of glutamate. That's the neurotransmitter that activates those NMDA receptors.
Okay, so it's like they're controlling the supply lines, adjusting the amount of glutamate that's available to trigger LTP.
You got it. And remember, those CB1 receptors are strategically located on both the sending and receiving ends of the synapse. So cannabinoids can influence both the release of glutamate and how sensitive the receiving neuron is to that glutamate signal.
Wow, so they're really controlling things from both sides. But I'm curious, do different cannabinoids have different effects on LTP? We've talked about THC and CBD having pretty distinct properties, so I imagine their influence might also differ.
You're absolutely right. Different cannabinoids can have varying effects on LTP depending on the specific cannabinoid, the dose, and even the brain region being studied. It's not a one-size-fits-all situation.
Okay, so it's more nuanced than just saying all cannabinoids either enhance or suppress LTP.
Exactly. For example, some studies show that high doses of THC can actually impair certain types of memory formation, and this could be because it's messing with LTP.
That's interesting, and it kind of makes sense considering some of the cognitive effects we associate with THC, like short-term memory glitches.
Right. But what about CBD? Well, its effects on LTP are even more complex and not completely understood. Some studies suggest it might actually enhance certain aspects of LTP.
Wait, so CBD could actually boost our learning and memory? That's surprising, considering it doesn't have those same psychoactive effects as THC.
Exactly. It just goes to show how complex these interactions are.
This is fascinating stuff. It seems like there's still so much we don't know about how these compounds are interacting with our brains. But speaking of complex interactions, what about synaptic pruning? We talked about how important that is for refining brain circuits. Do cannabinoids play a role there as well?
That's a great question, and the research is still emerging, but there are signs that cannabinoids might be influencing synaptic pruning too.
Wow, so not only are they shaping the formation of new connections, but they might also be influencing which connections get pruned away. It's like they're the architects of our brain circuitry, both building and remodeling at the same time.
A perfect analogy. And think about the implications. Synaptic pruning is crucial for refining brain circuits, eliminating unnecessary connections, and strengthening the important ones. It's like decluttering a room to make it more efficient.
Right, so if cannabinoids are influencing this process, they can have a lasting impact on how our brains are wired, both for better or for worse.
Precisely. And that's why it's so important to understand the impact of cannabinoids, especially during those critical periods of brain development. We need to know how exposure to these compounds might be influencing that delicate balance of synapse formation and elimination.
That makes me think about adolescents, teenagers. Their brains are still developing, so could exposure during those years have long-term consequences?
That's a very important question, and it's one researchers are actively investigating. There's growing evidence suggesting that heavy or chronic cannabinoid use during adolescence might be linked to some subtle changes in brain structure and function.
So it's not just about the immediate effects like memory lapses or altered perception. There could be more subtle, long-term consequences we're only beginning to understand.
Exactly. And that's why responsible use, especially during those crucial developmental years, is so important. We need to weigh the potential benefits against those potential risks.
This is giving me a lot to think about. But before we get too focused on the risks, what about the potential benefits of cannabinoids in this context? Could their influence on synaptic pruning be harnessed for good?
That's a fascinating question. The research is still in its early stages, but there are some intriguing possibilities. For example, some researchers are exploring whether cannabinoids could be used to promote synaptic pruning in conditions where there might be too much synapse formation or not enough pruning.
So it's like using cannabinoids to fine-tune those pruning shears, making sure they're working properly to create those efficient brain circuits.
Exactly. It's a bold idea, but it highlights the potential of these compounds to target those fundamental processes of brain development.
This is definitely mind-expanding stuff. We've talked about LTP, synaptic pruning, and how cannabinoids could be influencing them, but I know there's even more to this story, right?
Absolutely. Let's shift our focus to another crucial process, receptor trafficking. Remember, it's like that cellular postal service, making sure those important receptors are delivered to the right place at the right time.
Right. It's like those AMPA receptors we discussed. They need to be shuttled to the synapse to make those connections stronger. So are cannabinoids somehow messing with the delivery of those receptors?
Well, the research is still unfolding, but there's growing evidence suggesting they might be.
Okay, so it's not just about neurotransmitters and synapses. Cannabinoids might also be tweaking the system that controls where those receptors end up. It's like they're the ultimate puppet masters of neuronal communication.
That's one way to put it. And this influence on receptor trafficking could be a big deal for how our neurons function and adapt. By adjusting where those receptors end up, they could be strengthening or weakening specific signaling pathways.
This is getting incredibly intricate. It's like cannabinoids are orchestrating a whole ballet within the cell, controlling where those receptors go and when. But what are the implications of all this? How does it actually affect brain function and behavior?
Well, that's what researchers are trying to figure out. But some believe that this ability to modulate receptor trafficking might be part of why cannabinoids show promise in treating things like chronic pain, anxiety, even neurodegenerative diseases.
So by fine-tuning the delivery of those receptors, they might be able to dampen pain signals, reduce anxiety, even protect neurons from damage.
Exactly. It's a really exciting area of research.
This is truly mind-blowing stuff. I'm starting to see how these tiny cannabinoid molecules, both the ones our bodies make and the ones we might consume, can have such powerful effects on our brains and bodies.
And we're only just beginning to understand the full scope of their influence. There's a whole world of possibilities still to be explored.
Well, I'm definitely ready to dive deeper. What's next on our cannabinoid adventure?
Let's delve into the fascinating world of synthetic cannabinoids. Remember, those are the ones designed to target specific parts of the endocannabinoid system with even greater precision.
Right, those designer molecules that are like fine-tuned keys for specific receptors. I'm definitely eager to learn more about those and their potential.
Then let's jump right in. So, we were talking about ionotropic receptors, those fast-acting gates that directly control ion channels.
That's a quick on-off switch for the neuron's electrical space.
Exactly. But now let's talk about the other major category, metabotropic receptors. These are a bit more complex, more like those elaborate Rube Goldberg machines.
Okay, metabotropic receptors. The name makes me think of metabolism, but I'm guessing it's not that straightforward.
You're right, it's not. Instead of directly controlling ion channels, they kick off a whole chain of events inside the neuron. When a neurotransmitter binds to a metabotropic receptor, it sets off a cascade of intracellular signals, often involving these things called G proteins and second messengers.
G proteins and second messengers, those are new terms for me.
Think of G proteins like tiny switches inside the cell. When a neurotransmitter binds to a metabotropic receptor, it flicks that switch, activating the G protein.
Okay, so the neurotransmitter sends a signal, and the G protein springs into action. What happens then?
Well, once activated, that G protein can interact with other molecules, setting off a cascade of events that ultimately lead to changes in the neuron's behavior.
So it's like a chain reaction, a domino effect, all triggered by that initial neurotransmitter binding. But where do the second messengers come in?
Those are like the messengers within the cell, relaying the signal from the activated G protein to other parts of the neuron.
Okay, so it's a relay race, with the neurotransmitter passing the baton to the G protein, which then hands it off to these second messengers, and they carry the message to its final destination. What are some examples of those second messengers, and what kind of messages are they carrying?
One common example is cyclic AMP, or CAN-MP, which you might remember from our discussion about cannabinoids and neurotransmitter release.
Right, CMP. We talked about how cannabinoids can inhibit its production, affecting neurotransmitter release. So these metabotropic receptors can also influence CMP levels, but instead of inhibiting it, they might actually boost it.
Exactly. It all depends on the specific receptor and the pathway it activates. Metabotropic receptor activation can either increase or decrease CMP levels, leading to a variety of downstream effects.
Wow, so it's a whole intricate network of signals, with neurotransmitters, G proteins, and second messengers all working together to fine-tune the neuron's responses. What kind of effects can these metabotropic receptors have on neuronal activity?
They can modulate a whole range of processes, from opening and closing ion channels to regulating gene expression, even influencing the production and release of other neurotransmitters.
So they're like master conductors, orchestrating a whole symphony of cellular events.
That's a great way to put it. And understanding these intricate signaling pathways is essential if we want to truly understand how the brain works and how we can develop new therapies to target specific aspects of neuronal communication.
This is fascinating stuff. But I'm ready to go even deeper. What other aspects of neuronal communication are we going to explore?
Well, let's talk about how those receptors are actually organized within the neuron. Remember, those receptors are like tiny docks waiting for those neurotransmitter boats to arrive. But these docks aren't just randomly scattered, they're strategically positioned to ensure efficient and precise signaling.
So it's not just about having the right receptors. It's about having them in the right place at the right time, like a well-organized postal system for those neurotransmitter messages. But how does the neuron manage that? How does it make sure those receptors end up where they need to be?
Through a process called protein trafficking. Think of it like a cellular GPS system guiding those receptor proteins to their designated locations.
Okay, so the receptors are packaged and shipped with specific addresses, making sure they end up at the right synapse. What's the mechanism behind this?
Well, these receptor proteins, like many others in the cell, have specific sorting signals, like molecular zip codes, that determine their destination. And the cell has all this amazing machinery, motor proteins, adapter proteins that act like delivery trucks, transporting those receptors along specific routes.
So it's a well-coordinated delivery service, making sure those receptors get where they need to be. But where do those cannabinoids come into play here? Do they mess with this delivery system?
There's growing evidence that they might. Cannabinoids might actually be modulating receptor trafficking, influencing where those receptors end up and how many are available at the synapse.
So not only are they influencing neurotransmitter release and binding, but they might also be tweaking the very system that controls where those receptors are located. It's like they're micromanaging every aspect of neuronal communication.
A very apt analogy. And this influence on receptor trafficking could be a big part of how cannabinoids affect neuronal function and plasticity.
This is incredible. It's like those cannabinoid molecules are conducting a whole symphony of cellular events, controlling the movement and placement of these essential receptors. But what are the implications of all this? How does it translate into real-world effects on the brain and behavior?
That's what researchers are working hard to understand. But some believe this ability to modulate receptor trafficking might be key to the therapeutic potential of cannabinoids in conditions like chronic pain, anxiety disorders, even neurodegenerative diseases.
So by fine-tuning where those receptors are located, cannabinoids might be able to do things like dampen pain signals, reduce anxiety, or even protect neurons from damage.
Exactly. It's a fascinating area of research, and there's still so much to learn.
This whole deep dive has been absolutely mind-blowing. We've gone from the basic mechanisms of neuronal activation all the way to the intricate world of cannabinoid pharmacology. And it's clear that these compounds have a profound impact on how our brains work.
Absolutely. And as research continues, we're only going to uncover more about the therapeutic potential of these fascinating molecules.
Well, I think we've covered an incredible amount of ground today. From the intricacies of neuronal activation to the fascinating world of cannabinoids, it's been a truly mind-expanding journey. I feel like I have a whole new appreciation for the complexity of the brain and the potential of these compounds to unlock new therapeutic avenues.
I couldn't agree more. It's been a pleasure exploring these topics with you.
And to all our listeners, thank you for joining us on this incredible journey. We hope you've enjoyed this deep dive as much as we have.
Until next time, keep those brains firing.